vocabulary words that you're going to have to know. Genetics has a lot of them. So just accept it. There are a lot of words you're going to have to know. If you're a flashcard type person, you're going to be making some flashcards. A gene is the instructions to make something, usually a protein, and those instructions are written in DNA. We'll talk in lab today about how they're written in DNA. But the key here is the A's and C's and G's and T's we talked about before. Those are the actual information. It's the sugar phosphate backbone. That just holds it all together. The actual information is the A's and C's and G's and T's. Bases. All right. Next word. Well, you know what a gene is now. So a genotype is the type of genes you have. That's not hard, right? The genes you have. A phenotype is the traits you actually display. Think of what's physically expressed, or what you can feel, what you can see, things like that. So your phenotype is what you display. For example, we already did a problem. Do you agree that everybody did a problem? We talked about squash, and we had yellow skin ones, and we had white skin ones, right? Well, the white ones, their genotype was r, whatever, too little. So we would call it homozygous recessive, meaning only one kind, and it's the little recessive. But what was their phenotype? White squash. It's white squash. It's nothing complicated. The phenotype is their actual physical trait. Phenotype just describes what they look like. So if we're talking about the skin of a vegetable, it's the skin of the vegetable. You describe the color. Does that make sense? Phenotype is what's expressed. All right. Wow, we got new words. They said there were a lot. A genetic trait is anything that you can see or detect. If it's something that everybody has the exact same version of the gene, we say it's invariant. If it is something where everybody, there are different versions, and it's variable, we just say it's variable. That's your phenotype. So there are variable genes for hair color or eye color. Some people are brunettes, some people are redheads, some people are redheads from the bottle, some people are blondes, If we say you express a trait, that means that you have it and you show it. So this has a purple gene, do we agree? And it has a white gene, do we agree? Right? So we have two different types. This will be down in the slide. We call that a heterozygous. It has two different kinds of gene, two different versions of the same gene, I should say, two different alleles. And we're not seeing it in white. White's not being expressed. Purple is being expressed. The dominant is expressed. Does that make sense? Yeah. All right. So the genotype has the purple gene and the white gene. The phenotype, what you actually can see, is just the dominant. Good? All right. So, wow, you knew this one already. If you have a diploid cell, it's got two copies of genes, right? Two copies. Normal cells are diploid. We have two copies of each chromosome. We call them a homologous pair. I hope that sounds real familiar. Okay, so the homologous pair would be one from Pa and one from Ma. One paternal, one maternal. Humans have 23 pairs, 46 total. Most species, different numbers, different species will have different numbers. If you're something incredibly weird like the Chinese water deer with the fangs, then you might within one species have different numbers of chromosomes between males and females, or different ones in different populations, and you look at the people who write textbooks and say, they just do what they want. Okay, so haploid cells. Haploid reminds you of half the normal amount of DNA. Do we agree? Sounds familiar. Somatic cells are diploid. Reproductive cells like gametes are haploid. One set of chromosomes from each parent. And so you get a set from dad into sperm, you get a set from mom in the egg, and each one of those is haploid. Like I said, I usually write it one in to make it really clear, but the textbooks usually write one in. Usually just write N, right? If you see N, it means one. All right, and these are made through meiosis, not mitosis. Somatic cells are made through mitosis. Good? Good? Easy enough. All right, so here's a picture. Here's a picture of a person, and she's got two different copies, two different chromosomes. same chromosome, one from Ma, one from Pa, do we agree? The maternal one they made pink, nicely color-coded in the blood. The maternal one they made blue. In real life they just don't look like that. Black and white, or there's things red and purple. But anyway, the point is, these chromosomes are the same. They have the same genes on them. Would you agree that they both have an A gene? Would you agree they both have an E gene? Would you agree they both have an H gene? Well, we're getting there. But they both have some form of H, some form of E, right? Here's the thing. You can have two that are the same version of the gene. Right there we do. We have two dominants of the A gene. You could have two, we would say that she is homozygous, dominant for the A gene, whatever A is. Do we agree? Here, we've got a dominant and a recessive. So we would say she is heterozygous for the D gene. She has more than one kind. Do we agree? Here, for the H gene, she's also heterozygous. She has two different versions, version 1 and version 2. But they're both capital letters, which means what? They're both dominant. This is the way that your blood typing works. A and B, we'll talk about this more, are both dominant. This is what we call a codominance. Okay? They're both dominant to O. Good? What? Oh, so you have a type O? Okay. Good band, right? Type O negative. Come on, you're with me, Cameron, right? Remind me in lab. We can do that. Okay. So, more nerd words for genetics. Allele is a version of a gene. So, you would agree that right here in this drawing we had two of the same allele, the dominant allele. Right here we had a dominant allele and a recessive allele, but they're still the same gene. Anytime you are doing things that are still the same gene, it has to be the same letter when you do genetics. That's one of the mistakes some of the people made in one of the classes when they were doing their genetics problems. They're like, oh, well, the white skin squash and the yellow skin squash, well, I'll use W and Y. No, they have to be the same letter. They have to be the same letter because they're the same gene. It's just different versions of the gene. One is going to be dominant, that was the yellow. One's going to be recessive, that's the white. Are you with me? Okay. So that's a really important thing you have to get, is that you have to use the same letter. And usually when I pick letters, because you can pick them however you want, I try to do something where I don't like having, oh, well, it's big P and little p, where the big P means it's a purple flower and the little p means it's white. That's just confusing. I'm going to go with C for color. And then the big C means purple, and then the little c means white. And then you're not confused by the P purpleness, you know. But you can do whatever works for you. Just avoid X and Y, unless we're talking about sex chromosomes. Good? Good. All right. So let's talk about versions of a gene. Would you agree that there's a version of the hair gene? I know this is a little oversimplification. There are multiple involved. But there's a version that gives you blonde hair. There's a version that gives you redhead hair. A version gives you brunette. A version gives you black. These are different versions of the same gene. Do we agree? Yep. All right. Genetic diversity. It's involved. All of these, I should have put them in bold and red, but you get the point. You know these words. Genetic diversity is having more than one version of an allele in a population. We usually think that the more, well, definitely, the more alleles you have, the more genetic diversity. We usually think this is a good thing, because if you're too inbred and you have all the same genes, you might end up looking funny, like British royalty with big ears and buck teeth and bleed a lot, like hemophilia. But it could be worse. It could be like the king cheetah, and now you're weak, and then when the coming plague, you don't have any genetic diversity, you're all going to die. Does that make sense? If you have some diversity in the population, you might lose this guy and this guy, but that guy's going to make it, because they're not all the same. Good? So that's why diversity is considered to be a good thing. All right. Hey, we did this before. A phenotype is the type of genes that you're actually expressing, the physical traits. Good? The genotype is the type of genes you have. Now, within genotypes, we have different names you have to know. Homozygous and heterozygous. If you have all the same type of genes, all the same versions, you're going to be homozygous. Remember I said you started with this purple plant? Thinking you had purple parents and purple grandparents and purple great-grandparents? It's a pretty good guess at that point that they're all because you bred out anything that wasn't, right? So that means if you got 2 copies of the purple gene, you are a homozygous for the purple gene. You're homozygous dominant, in this case it's purple dominant white. Good? Good. So homo means the same. Hetero means not the same, right? This is also a purple plant, but it is heterozygous because it's got a gene from the purple plant, it's got a gene from the white plant. 2 different kinds, not the same, so it's heterozygous. Are you with me? All right. So a genotype, we can talk about something saying it's a hybrid. When we say hybrid, it just means that this strain got bred to this strain and you're now a heterozygous. That's a hybrid and a heterozygote are kind of the same thing in genetics. Good? It doesn't have to mean this species and this. It just means that family, that strain, that strain. Dihybrid. Hey, what's di? Would you agree we got the S gene we're talking about and we got the T gene we're talking about? All right. And would you agree that it's hybrid for the S and it's hybrid for the T? That's why it's a dihybrid. In this case, this is a famous one. We'll talk about this experiment a lot later. But Mendel went and he bred round, smooth peas that were yellow with wrinkled peas that were green. And so he had a skin texture gene and he had a skin color gene. And in the offspring, they had the dominant for both. Yeah. But we'll talk about the fun thing, what happens when you breed them with each other. How many of you guys looked at the magic ratio sheet that I said to look at the magic ratio sheet? Oh, it's got it. So, you mind a hard question? All right. So, if I told you that the offspring of a cross had a ratio of phenotypes of 9 to 3 to 3 to 1, what do you know right away?
I glanced at it.
You glanced at it,
Oh,
okay.
okay.
Well, if you had studied it in depth, and by the test you will know
Yes.
this, if you see a 9 to 3 to 3 to 1 ratio of phenotypes, you know that there are 2 genes involved. You know that both of the parents had the exact same genotype, and you know that that genotype was dihybrid heterozygous plateau. You know all of that just from 9 to 3 to 3 to 1. You don't need to make a 16 square Punnett square and muck around. Well, I can't read that, it's too small, is that a big deal or a little bit? None of that. You know the answer. That's why the magic ratios are magic. If you memorize that sheet, you can walk into any classroom in an introductory biology class in the US and answer at least 80% of the questions without ever doing the Punnett square. Go learn the sheet. Moving on. Okay. Dominant is an allele, a version of the gene that will be expressed if it is there. Have you heard that? I told you before we even got there because I wanted to make sure you knew it. I said you had to write it down. Show me why. Oh, it's so on the test. It is so on the test. The reason we're using my definitions is because mine work both complete and incomplete. If you were to use a book definition, you'll be, well that works except here. And screw that. This is always true. Always true. This is always true. The recessive is an allele that will be expressed if it is not being blocked. So let's look right here again. We got a dominant. Would you agree? If it is present, it will be expressed. My God, it's expressed. You got the purple flower. All right, what's our definition for recessive, Grace? Don't look. Exactly. All right, so you have the gene. Do we agree? This flower has the gene. It's being blocked. You don't see it. Right? That's why we know it's a recessive. There's one other thing. You could have multigenic traits. Like human skin. There's not like one gene for human skin color. There's like 6 at least. Some people think there might be as many as 12. We're still figuring that out. And that's why we might as well jump to the exciting bit if you want to see what that looks like with just 6 genes. Flipping in your book. Look on page 294 in your textbook, those of you who are clever with my textbooks. Yeah, I did say that. I know, you're like, I never read my textbook. That's okay. How did they do that? I didn't fix it. I know how they did it. I don't know why they did it though. Alright, so if you had 1, 2, 3, 4, no, they use 7 genes here. Like I said, some people think you might have up to 12. You would agree, it's not like you have really light skin or really dark skin. There's a whole palette all the way in between, right? The reason why is because you have, in this drawing, 7 different options from dad and 7 different options from mom. That's a lot of variation. Do we agree? That's what we're talking about. That is a multigenic trait. Are you with me? And probably it should say it doesn't say pleo or pleiotrophic, but the next one, we'll get to it eventually, is the opposite, pleiotrophic, where one gene controls multiple traits. Okay? Just write it down. You'll see it eventually. You'll have to write it down on that slide. If you want to wait, you can write it down then. But yeah, that's another one. All right, so let's talk about this. I already said this, but you have to use the same letter if we're talking about the same gene. If one version of the gene makes the squash yellow and one version of the gene makes the squash white, they use the same letter. If one version of the gene makes the flower purple and another version of the gene makes it white, you use the same letter. Does that make sense? Because it's the same gene. The way you tell the difference is not the letter, it's whether it's a big one or a little one. We use capital letters for the dominant version, the dominant allele. We use lowercase letters for the recessive. You got F1, F2. A gene is the instructions to make something that's usually a protein. Do we agree? Yep. All right. And do you have lots and lots of different genes on a chromosome? Do we agree? All right. Good. A genetic cross is any mating experiment often performed to look at the inheritance of a particular trait. We'll talk about a special example of that called a test cross later. It's something that's done in horse breeding. All right, the genotype's the type of genes you got. The phenotype's the type of genes you are actually expressing. The ones that are actually going to affect the phenotype. Do we agree? Because it is possible to carry a gene without expressing it. If you do that, would you be homozygous dominant, homozygous recessive, or heterozygous? Heterozygous. Heterozygous. All right. Heterozygous means you have 2 different alleles of a gene. It's the same gene, but it's 2 different versions, right? Homozygous means you have 2 of the same version, the same allele of a gene. The P generation is the parent generation. When you're making your cross, when you first bring the 2 things from different traits together, that's the P generation. Phenotype are your actual physical traits that you have, your height, how fast you can run, things like that. Your recessive allele, we already did that, and a trait is any feature that we can observe or detect. Are we good? Yeah. So all these terms, we've pretty much gone over them, but if you don't know them, you're going to have to know them. They're in your book. They're table 12.1. All right, so Mendel loved peas. He liked peas a lot. He worked with them for years, 7 years. All right, so what he did is he took a purebred, true breeding purple plant. And he knew that its parents were purple, and its parents were purple, and their parents were purple. Had nothing but purple genes. Leah, what's the fancy H word for that? Homozygous what? Dominant. Very good. All right, so we have a pure purple and we have a pure white, a homozygous recessive, and we breed those together and we get, Daniel, what's the H word?
Heterozygous.
Excellent, heterozygous. All right, and you notice the heterozygous is exactly like the dominant, has the exact same phenotype, looks the same, has the gene for the recessive, but it doesn't look that way because the dominant is completely blocking the recessive in the plants that Mendel looked at. Mendel got lucky. Nothing that he looked at had incomplete dominance. We now know about incomplete dominance. You know about incomplete dominance. He didn't know about that stuff. Good? Now, if we take
this, and we breed it with its sister. I know that's kind of creepy, unless you're British royalty, they're used to that kind of thing. But if we breed it with one that is also a heterozygote, sorry, Anthony, just speaking the truth. How do you think they got buck teeth and big ears and the tendency to bleed a lot? Recessive traits, inbreeding. Yeah. All right. Anyway, so you take a heterozygote, you breed to a heterozygote, and you get a 3-to-1 ratio. Yeah.
What chapter is this?
This is, I think, 12. Is that correct? What?
Oh, never mind, it's test 4, not chapter 4.
All right. Yeah, he's right. It's test 4. I think it's 12, but the important thing is it's Mendelian genetics. If you see a 3-to-1 ratio right there, how many people have looked at the magic ratio sheet that I said memorize the magic ratios?
Scotty, you glanced at it.
Okay. Well, nobody else memorized them. If you memorize those ratios, you know everything there is to know about a problem. If I showed you just this right here, and you saw that the children have a 3-to-1 phenotypic ratio, you right away can tell there's one gene involved. You right away can tell that both parents are heterozygous for that gene. You right away can tell that that gene is completely dominant. You right away know which one is the dominant and which is the recessive. All that information's right there. Okay. So Mendel, when he did this, he did it with 7 different traits, and every time he looked at one of them, he saw a 3-to-1 trait. Okay, 3-to-1. Like I said, when you see the 3-to-1 ratio of phenotypes in the offspring, it tells you there's one gene. It tells you that one of those alleles is completely dominant to the other allele. It tells you both parents are heterozygous. Are we good? Yep. There's the little Punnett square. All right. So Mendel had 5 big ideas. These were new ideas to him. Of course, nobody noticed them for 30 years, but they were big ideas. All right, so Mendel's important ideas. There's a variation in traits. There are different versions of a gene. Sheldon, there's fancy A word for those versions. What do we call them? I'll try that again. You awake? You with me?
Barely.
What do you call a version of a gene? I'm looking for an A word. Help him out, Cheyenne. Oh, say it loud so everyone can hear. Don't whisper. Yeah, allele. Very good. Very good. You're going to know that by test time, right?
Yes.
Excellent. All right. Mendel came up with the idea that there are different versions of genes. Well, that's a modern way of saying it. He knew there were he knew there was information that was passed on about a trait. And he knew that there were different versions of that information. And he knew that you got a copy of that information from each parent. And he knew that it was possible for one to block the expression of the other. Before, people were like, oh, you breed a white cat, you breed a black cat, you get all gray cats. I don't know why they thought that, because you never have. It's never worked that way. But he's like, you breed purple pea plants and white pea plants together, and what do you get? The purple, because it blocks the white. That was a big deal. Nobody knew that before. And it wasn't just, he didn't just with flowers, he did it with 7 different traits. One of them would have been skin color. You breed the yellow peas and the green peas together, and all the offspring come out yellow. The reason why is that yellow is dominant. It fully blocks the expression of the green. Does that make sense? Yep. All right. 2 copies of a gene got separated during reproduction in preparation for reproduction. They ended up in separate gametes. And he's the one who, well, it's random fertilization. It happens randomly. You never know what the offspring is going to be. It could be this way, it could be that way. Remember our little sperm that was wearing the mining hat and it got lost? Yep. All right, so Mendel really liked peas. 7 years, 29,000 plants. We already talked about how he bred them, how he would put a little cover over each flower, and then when it was time to breed that flower, he would take it off, and he would take another flower and get the stamen with the pollen off of it and dust it, and put the little cover back on so you couldn't blow any pollen on and fertilize it for some other plant. So his experiments were tracking one trait. He did 1 and 2 traits. So he did. All right, so the law of segregation was something he came up with. And for law, I want you to put like quotes around that or something. I want you to make sure that you understand. He described what he thought was a law. It's more of a suggestion. What he said is use usually true, but he was lucky. Everything he looked at was on separate chromosomes. If you have two different genes for two different traits and they're on the same chromosome, they might be more likely to go together and not do independent assortment. Does that make sense? Mary says no. So let me try it again. Put one hand down. You put yours up. Remember I said they can go in different directions and all that? They could line up like this, or, now trade places. Or they could line up like that. Do we agree? They line up on the metaphase plate. They can line up either way during metaphase 1, right? That difference, if we're dealing with separate chromosomes, then the genes will move separately. But suppose you have one gene on your wrist or one gene on your ankle. You're still chromosomes, okay? But you got one gene on each. Would you agree they're more likely to move together than to do a crossing over? Like, they're not wanting to touch each other. You see that? Do a crossover.
What is that? Like,
Yeah. Okay. You see how unlikely that they're going to get a crossover? Are you with me? Okay. Those genes are going to move together more often than they cross over. Does this make sense to you guys? Because they're on the same chromosome. Mary, are you with me now? All right. All right. So Mendel thought he had the law of segregation. It's more of a suggestion. All right. So a Punnett square shows all the ways that you could have things combined. That's the famous Punnett square. That's the one you're going to tattoo on your wrist, right? No.
Do we have to know that one particular?
Well, you have to know a 3-to-1. I don't care what letters you use. It could be an E or a P or a Q or whatever makes you happy. No. The letter is not important. What's important is the Punnett square. As long as you use the same letters for the same gene, your Punnett squares are fine. I would avoid X and Y, Z and W because they're used in sex determination. But other than that, use whatever letters you want. I personally would not have used P like the book did here because they're using big P for purple and little p for white. And little p for white to me is confusing. I would have used C for color, and that works for both, you know. The dominant color, the recessive color. That's just me. All right, so if you see a 3-to-1 ratio, if there's a 3-to-1 ratio in the phenotypes of the offspring, you know there's one gene involved. You know that the allele has complete dominance. One of the alleles has complete dominance. You know that both parents are heterozygous. You've heard this before. You're going to know it by the test. Do we agree?
Si, senor.
Yep. All right. So Mendel, more P's, please. Right. P's. Okay. So he's looking at more P's. This time he looked at 2 traits, not 1. He looked at seed color and seed texture. You never thought about when you eat peas, those are seeds. It's a dihybrid cross because he's looking at 2 different things. And the parents in both, what he did is he took parents that were heterozygous for both traits. So they had a dominant and a recessive for whether the thing was round. They had a dominant and a recessive for whether or not the thing was yellow. So texture and color. Use an R for the texture. See, I would have used T. They're using Y for the color. See, I would have used C. Anyway, but what he found was independent assortment. The copies segregate separately from each other most of the time. Like we just talked about, if the genes are on different chromosomes, works perfectly. He got lucky. Everything he looked at was on different chromosomes. We now know enough genes that he didn't that we know cases where that won't work. Does this make sense to you guys? So that's why, again, independent assortment suggestion. All right. This is the 9-to-3 to 3-to-1. If you see 4 different phenotypes, what does that tell you right away? There are 2 different genes. If you see 2 different phenotypes, what does that tell you right away? There's 1 gene, right? Okay, so 9-to-3 to 3-to-1. If you have 9-to-3 to 3-to-1, it tells you there are 2 genes. It tells you that both genes have complete dominance. It tells you that both parents are heterozygous for both genes. Memorize those things for those ratios. It'll make life really, really easy. Somebody bust out a calculator.
I got you.
All right. Okay, I'm going to get a marker to write with. Let's write some stuff down. I want you to add up the number of round yellow pants and round yellow and the number of round green. So add 315 to 108.
423.
Okay, so all the round are, you said what, 423?
Yeah, 423.
All right, 423. So what are all the wrinkled? 101 plus 32?
138.